Welcome back. In the first video of this 3-video series in the urinary system, I introduced the nephron and its location in the kidney. This diagram represents a nephron, and if you remember what I said, is it has a very twisted tubule that travels up and down and through, making 3 passes between the cortex and the medulla. So let me put some of these things down. This is the cortex. On that side of this imaginary line, this side is the medulla of the kidney, the renal medulla. This is where the finished urine gets put, down here. This tube goes to the renal pelvis. The contents here are technically outside of the body. So if we follow the tube, this is outside the body. Notice there's branches. We have these nephron structures. This is a nephron. There could be many nephrons connected to one of these things, which is called, quite simply, a collecting duct. And out here, of course, we could have a nephron. Another one here. This whole thing represents a nephron. And if we look at it, we can sort of describe the parts to it. There's a distinct structure here. This is a separate part. This big loop is another part. There's another part. And we call the collecting duct the final part. So if we look at it, we have part 1 here, which is known as Bowmans Capsule. Bowman is always capitalized because that was a person's name. We have this part here, this very twisted tube. It's the proximal convoluted tubule. Let me put that down here. Proximal convoluted tubule. I would often abbreviate it just PCT because that's quite a term to write down all the time. So we go from Bowman's capsule, proximal convoluted tubule, to this loop structure. This is known as the loop of Henle. Again, notice the spelling of Henle. This was a person's name, so it's always capitalized. And there is a descending and an ascending portion of the loop of Henle. And we have another thing that looks very similar to the proximal convoluted tubule, particularly the way I drew it here. This is the distal convoluted tubule. And the collecting duct. So in terms of the numbers of structures, 1, 2, 3, 4, 5. And the process of urine formation, the formation of urine, which is the topic of this video, has a part of the story occurs in all of those sections, and we have to get into those. So there's stories at least 5 parts long. Before we get to that though, we have to get back to where we represent the filtration process, because all of this structure is representing the movement of material which is outside of the body. We haven't had the filtration process yet. The filtration process is what occurs in here. So let me add some blood vessels to this, because we know that the renal artery brings blood in. By the time we get down into this level, this microscopic level, there's millions of these in the kidney and they're very microscopic. We have to be talking about renal arterioles. So there's a renal arteriole that'll bring blood in, and the blood actually forms a very delicate capillary-like knot of blood vessels right in here, right inside Bowman's capsule. and then the blood will go this way into here, and the blood will actually go out that way. Notice that I'm still coloring it red because this is all arteriole. There's been no exchange to take out the oxygen yet. This is not a venule yet. This is not on the other side of the capillary bed. This is just a knot of capillaries that's inside Bowmans capsule. Now, I have to think too that representing Bowmans capsule this way is a bit awkward because it doesn't really tell the truth. It's not just a cross section like that. It's a cup-like structure. So if you could imagine my arm here to be the proximal convoluted tubule and my hand to be Bowmans capsule, the blood vessel comes in here and forms that knot of tissue in there. And this thing actually surrounds that. So the structure in here has its own name. This is a glomerulus. Glomerulus. Obviously be glomeruli if we're talking about more than one. I mentioned that this is an arteriole. This is still an arteriole. Well, now that we've got two arterioles in the same region, we have to call them something different. We can't say, oh yeah, the arteriole. No, there's two of them. This one that takes the blood in is the afferent arteriole. This one that takes the blood out is the efferent arteriole. Notice that, again, these things are in alphabetical order, so if you ever have any confusion as to which one's which, afferent is first, goes into the glomerulus, efferent takes the blood out of the glomerulus. And obviously out on the end of here, we have our capillary bed. And that capillary bed it'll take the blood back to the renal arteriole, then into the renal venule, I should say, and into the renal vein. That capillary bed actually exists in the same location as the nephron. So it may be that the capillary bed itself wraps around the same nephron. So we could have a capillary bed that wraps around this thing here. It could wrap around the same nephron, it could wrap around other nephrons. That part doesn't matter, it's just the fact that the capillary surrounds the nephron. So we have this whole network of capillaries that surrounds the nephron, the loop of Henle. So the blood could come down into one of these capillaries, and the exchanges occur here, and that blood is taken away. This blood again goes to the renal vein eventually. So that's the lead into this story. What we have to do in this video is look at what happens at each of these places along this nephron structure. So we'll start by looking specifically what happens at Bowmans capsule, then the proximal convoluted tubule, and so on like that. But I've got way too much stuff here right now. I have to erase some of this and present it bit by bit. I'll be back in a minute. Welcome back. So we're ready to start looking at the steps in urine formation. And like I said, there's 5 steps. The first of these occurs at Bowmans capsule. The blood, remember, comes in the afferent arteriole. It forms a glomerulus, which is a set of delicate little blood vessels that are encapsulated by Bowmans capsule, and the blood goes out on the efferent arteriole. We're dealing with arterioles here. So the amount of blood pressure in here might be somewhere in the range of, say, 40 millimeters of mercury. It can be higher than that a little bit sometimes. at the beginning of a capillary bed, so off here where it forms the capillary bed that goes around the nephron structure itself, we're dealing with blood pressures of 35 to 25 to 15. Well, here we're a little bit higher than that. This blood pressure is sufficient enough to actually force material out arterioles here. Just the direction of this, the blood pressure's forcing that way, pushing that way, as this knot of tiny little blood vessels is encapsulated by this Bowmans capsule. So it literally happens because of the blood pressure. Fluids get forced out of the blood and they can't go anywhere but into Bowmans capsule. The fluids that get forced out are in the tubule, outside of the body, because the components that are in the tubule are outside of the body. So there's our filtration process. That's how we get the waste materials out. So what happens, quite simply, this stuff is our filtrate. So the filtrate enters the tubule. But we have to examine exactly what's in the filtrate, because believe it or not, it's not all waste material. We have two kidneys, and if they're both fully functioning, they say that the kidneys filter about 180 liters of fluid. There's 180 liters of fluid filtered every day. Well, our body only holds about 5 liters of blood. In terms of the math, for every 125 milliliters of filtrate that gets produced, we wind up with 1 milliliter of urine. So all of that material gets filtered out. This is about 99.2% doesn't wind up in urine. 99.2% winds up getting brought back into the system, but not at Bowmans capsule. Though that's the process that occurs further along. One thing we have to look at is what exactly is in the filtrate. What gets filtered out? The filtrate contains quite simply anything that's small enough to get filtered out. Certainly a lot of water, glucose, there's blood sugar, it gets filtered out. The urea gets filtered out. We have ions that get filtered out. That's just about it. Most of the stuff that's in blood is too big. Glucose and other food materials, I suppose, or other unit molecules, but most of the stuff that's in blood is too big to get filtered out. It stays in the blood. So the things that are too big does not contain cells, does not contain protein. So the blood proteins, like we have albumin, for example, is a blood protein that floats around in the circulatory system and helps create osmotic pressure. It's too big. It doesn't go through. Things like histamine is a protein that's released by white blood cells. Things that other large molecules. We just put it that way. Protein and other large molecules. So what you have to recognize is that the blood that's in the afferent arteriole, the blood that's in the efferent arteriole, is quite different. It's still blood, it's still arterial blood, it's still oxygenated blood, meaning the hemoglobin is still transporting oxygen, but this blood contains more water, more glucose, more urea, and more ions than the efferent blood. Less water here. This blood here, because it contains less water, is actually a little bit hypertonic compared to the blood that's in the afferent arteriole. And I point that out because that's a very important detail. That's the first step in urine formation. It's called pressure filtration. It occurs at Bowmans Capsule, it produces a filtrate that has things in it that we still need. We can't afford to lose all that water. We have to get the water back. We can't afford to lose our blood sugars. We have to get those back. We can't afford to lose all the ions that are floating around in blood and keeping us healthy all the time. We have to get some of those back as well. But that's the first step. We have to make the filtrate. We have to start the process to get rid of the urea. The second step in urine formation occurs along this next tube. Bowmans capsule. This tube is the proximal convoluted tubule. And the reason it's called proximal, if you think about the term approximate, if it's approximately correct, it would be close to correct. Proximal just means that it's close to Bowmans capsule. So if we recognize, here's Bowmans capsule out here, I'll just put it like that. And we've got the proximal convoluted tubule that leads out of Bowmans capsule. And I said I abbreviate this oftentimes just PCT, proximal convoluted tubule. The cells along the proximal convoluted tubule are actually wrapped around with the arterial blood. So we have blood vessels that are traveling very close to here. And they may be branching, the arterioles are leading into the capillary bed. In fact, these can be capillaries as well, they're very, very delicate. The cells of the proximal convoluted tubule, the cell walls are only one cell thick, and they have specializations in these cells, so there's specializations along the cells, along the tubule here. Those cells are specialized to transport things that we need back into the blood. So things like the glucose. Glucose would be inside the filtrate in here. Remember, the filtrate gets put down from Bowmans capsule. And these cells will actually push things out of the tubule into the tissue spaces. A little bit of water, a little bit of sodium, not much, but some. A little bit of chlorine follows passively. So we do get some of the ions back, but it transports these materials into the tissue spaces of the cortex. Now comes the point that I made before. The blood that's inside this blood vessel here, the arterial blood, is hypertonic. Because it's hypertonic, that sets up an osmotic imbalance, and materials which are in abundance in the tissue spaces here, these things here, the water and glucose and so on, get transported back into the blood. So we recover them. It goes back into the blood. So we don't urinate out our glucose. Our glucose is almost 100% reabsorbed right here because we need to get our glucose back. We can't lose all that water. We can't lose all the ions. So we have this process which depends on the presence of specialized proteins in these cells to transport the glucose, water, and ions into the tissue spaces so it can get drawn back into the blood by osmosis, course, the osmosis pulls the water, and things that are in the water can go along with the water. So we get that back into the blood. This process here is called selective reabsorption. And it occurs along the proximal convoluted tubule, and it reabsorbs glucose, a little bit of water, some ions, and things like that. Notice there's no urea. Urea is part of the filtrate. It stays outside of the body. It stays inside the tubule, and in fact, with the removal of that little bit of water, the urea begins to become a little bit more concentrated. Most of it's concentrated in the next part of the nephron, and the next part of the nephron following the proximal convoluted tubule is the loop of Henle. And remember, the loop of Henle drops from the cortical region down into the medullary region of the kidney. So here we have the proximal convoluted tubule. Now we have the loop of Henle. And let me just represent cortex versus medulla again. Cortex on the outside, medulla down here. This whole business of selective reabsorption no longer occurs when we get down here. The reason it doesn't occur in the medullary region is the concentration of sodium is quite high. So the whole process has to be shifted a little bit. The fact that it's high and the fact that these cells that line these tubules are equipped with carrier proteins, transport proteins that allow water out, and you have a high concentration of sodium here, water leaves. We get a lot of water taken out of the descending side of the loop of Henle. Remember, the flow of filtrate goes this way, goes down here, so we say it's descending, it's going down the loop of Henle. This is the descending side. The ascending side is different. So a lot of water gets withdrawn on the descending side of the loop of Henle because of two features. Because of the transport proteins that are in the cells that line the tubule, and because of the high sodium concentration here. This process here I simply call water reabsorption. So that's the third step here. So this occurs along the descending side of the loop of Henle, and it allows water into tissue spaces. We have to get the water back into our blood. It's no good just floating it around in the kidney. So again, what happens is very simple, that the blood vessels continue to wrap around the nephron loop of Henle here, and that osmotic gradient exists where the water being present in the tissue spaces, and the blood is still hypertonic because it's still lost all that water out here. This water, it's the same water, will get drawn back into the blood. So there's actually two movement processes involved here, where we get the water out of the tubule and we get the water back into the circulatory system. Okay, now for the distal side here. Distal side, which is made up of the ascending side of the loop of Henle and the distal convoluted tubule, which is up here. The fourth step. The fourth step in this whole process I call sodium recovery. You see, what's happened quite simply is we've got weak urine up here. Weak urine that contains sugar and some other things which are important. The sugars and the other things that are important are withdrawn. So this is weak urine. The urea has we haven't done anything to urea. It's still here, and so are a lot of these ions. So basically what simply happens down here is this filtrate, by the time it's down here, a lot of water has been taken out of it. It's quite concentrated with urea and ions. Remember, there are a few ions taken out up here, but there's still a lot of ions down here. So what we actually say is that the filtrate becomes quite salty by the time it gets down here. This is quite salty. The concentration of ions is quite high. So what has to happen is we have to recover some of those ions. Now the specializations on this side of the tube allow that to happen, because in here, let me just put it down, we have salty urea, or salty urine. It's not really urine yet, it still has to be processed somewhat, but we have to get rid of some of the sodium from here. Now, at the very bottom of this loop of Henle, where the sodium concentration is quite high in here, it's not going to pass on its own quite realistically, is it? If it's really, really salty, and if this salt concentration exceeds this, yeah, you can have some diffusion of sodium leaving, as long as the proteins are there, leaving the tubule. But as you go up here, it gets less and less and less salty, or the concentration of ions decreases. You actually have active transport kicks in to remove some of the sodium ions. But I have to mention one thing first. These water channels only exist on this side. If they existed on this side, you'd have the water being drawn back in, and we don't want that. The structure of the descending side and the ascending side are totally different. These have protein channels which allow water transport. These have proteins that allow sodium transport. And of course, chlorine ions follow passively. So what happens on this side is that the sodium ions leave the filtrate through these cells which contain the proteins to allow it to be transported. And as long as the concentration difference is sufficient, it can pass out passively. But as the concentration in here, as we remove sodium ions, and the effect of sodium concentration diminishes as you go up here, it actually is actively transported out. One other thing before I make a note behind me here. Notice that sodium. The sodium concentration which draws the water out is because of the sodium that gets pumped out and moved out on the descending side. And of course, it stands to reason that we have millions of these nephrons. So it's not just one, there's another one here and another one here. So this whole high sodium concentration nature of the medulla is created by sodium ions that are moved out from the ascending side of the loop of Henle all over the place. So sodium recovery, it occurs in the ascending side of the loop of Henle, and it depends on the presence of sodium transport proteins, notice as well, I have to make this comment, that the ascending side does not permit water movement. simply because the structure of the cells is different. They don't have the cells that have the protein channels in them, so the water, it's stuck out, which is good. Another thing I should comment on, I'll put a fourth point down here, is that by the time this filtrate gets up here, it's not so salty anymore. The salts are being removed, the ions are being removed, sodium, chlorine being removed. So what we have, we're concentrating urine. We have water removed here, it's salty, now we get the salts removed, so what we have up at this point is almost urine. It's way more concentrated because all the water came out, and it's way less salty because a lot of that, not all the water, but most of the water, and a lot of the sodium ions and chlorine ions came out on this side. So this is not so salty anymore. In fact, this is literally isotonic to this. So this material in the distal convoluted tubule and the proximal convoluted tubule, in terms of their ion-water ratio, they're isotonic. We've just lost a lot of water and a lot of salts. So the fourth point is by the time you get distal convoluted tubule, by the time you get the filtrate up to the distal convoluted tubule, it's less salty. So now comes what happens in the distal convoluted tubule. And I should mention the term distal. I did with proximal. Proximal, if you say it's approximately, it's close to. Distal, at a distance. So the distal convoluted tubule over in this location here is at a greater distance from Bowmans capsule. Let me erase this picture and just represent the distal convoluted tubule. So we have the loop of Henle, the ascending side of the loop of Henle, the distal convoluted tubule, which leads to a collecting duct. I'll represent another nephron entry point there. So this distal convoluted tubule. The other feature I can add to it is the blood vessels. And now at this point we actually have, I'll still represent it with blood, with red, to distinguish it from the tubule, but the blood vessels here could very well be venules because of the capillary bed that wrapped around the tubule itself. So these blood vessels are leading off to the renal vein. The filtrate is drawn up here. Remember, the filtrate now is about isotonic to on the proximal side. It's more or less concentrated urine. What is in this filtrate here, for example, is very similar to here. This collecting duct, CD collecting duct, takes it to the renal pelvis where it's stored before it goes to the bladder. The features that these cells along here are specialized for is to help maintain some sort of homeostasis with the blood and the urine and so on. You see, blood is supposed to have a pH, for example, of around, supposed to be slightly alkaline, and it tends to become slightly acidic. So what normally happens is there's an exchange of hydrogen ions between the filtrate and the blood, or the blood and the filtrate, so that we can maintain the correct pH of our blood. But at the same time, there's another process too, and that is that there are things in the blood which were too big to be filtered out that we don't need, we have to get rid of. And I'll draw your attention to perhaps two of them. We have histamines. Remember, histamines are produced by a certain type of white blood cell in reaction to some sort of an allergy or an infection. It causes tissue swelling, and the histamines make the blood vessels leaky. Well, afterwards, the histamines are drawn off, and they have to get out of your body somehow. Histamines are too big to be filtered out at Bowmans Capsule. Same as penicillin. Penicillin, you take penicillin, an antibiotic, for example, it floats around in your blood. Well, what happens to it? Eventually you have to get rid of it. It's not there forever. It actually is removed from the blood into the nephron. So we get things like a movement this way of things like histamines, penicillin. We have ions, and I'll come back to the ions in a minute. And we have a movement from inside the tubule back into blood of primarily just ions, because we're trying to balance ion concentration. This brings me back to the point that I made at the very beginning, or almost the very beginning of the first video in this series. This movement of material from blood into the outside of the body, this is an excretion process. This movement here of things into the blood which are still useful, this is a secretion process. So what we have along the distal convoluted tubule is a process which is known as tubular secretion and excretion. Now, the ions, I said I'd come back to the ions. The ions, if blood pH is too low, that means that there are too many hydrogen ions in the blood. And the condition of acidosis could occur, denaturing proteins, and you don't feel well, your body doesn't function well. If the blood pH is too low, we have to excrete. If we have a low pH, we have to excrete hydrogen ions, which means quite simply the hydrogen ions are going to go along the same direction as the histamines and the penicillin. They'll be going into here, and urine will become more acidic. We also at the same time can absorb bicarbonate ions. Bicarbonate ions can be present or are present in the filtrate. So if we absorb the bicarbonate ions and excrete the extra hydrogen ions, that will raise the pH and bring things back to about 7.3, 7.4, which is more of a normal range for pH. Of course, if the pH of blood is too high, this would be exact opposite. If the pH of blood goes too high, we'll actually absorb hydrogen out of the nephron into the blood, and we'll excrete bicarbonate ions. So there's the ion exchange. So tubular excretion secretion occurs at the distal convoluted tubule. One of the most important things of it is it regulates blood pH. And it also allows the excretion of histamines, penicillin, large molecules. Things that were too big to get filtered out at Bowmans Capsule. So now for the last step in this whole process, and I call it the same as before. So it's the water recovery thing where we're retaining the water, we're concentrating the urine. This urine that we have at the beginning of the collecting duct here, it depends on what your body activities are, obviously. If you haven't had a lot of fluids, it may be fine. If you've had a lot of fluids, it may be weaker. So I mean, it depends on your body activity. But the collecting duct has to be designed in such a way that you can recover more water. And what happens along here is that there are more of those cells that contain protein channels, so that as we're moving this collecting duct, remember this is the third passage into the medulla, and the medulla has a high sodium concentration. As this goes down here, water can be removed. So we can remove additional water. So step number, I'll say it's step number 6, but it's really the same process as what was occurring in the descending side of the loop of Henle, where the tubule there is specialized with cells that have protein channels for allowing the water out. At the end of the day, like I say, 99.2% of the water gets recovered. We can't afford to lose it all. One last look at what's going on here, what's in here. This is the filtrate. Here, remember, it's come down to the nephron, or down the nephron, through the nephron, down the collecting duct to the pelvis. What it is a lot of urea, and it's basically our urine. The next video in this series is going to look at making adjustments, because your body is not acting the same way all the time. The body's needs for homeostasis, for maintaining homeostasis, are not absolutely constant. We do have some hormones that play a role in this in case you have to get extra water out. There's things that can go wrong with this system, obviously, and that's the next video. Thank you very much.